the introduction there.
So just for the people who don't know me, I'm Ursel. I run the HOLOBIONT podcast. And the reason I wanted to organize this talk today was to share this research topic, which I found very impressive, and I'm sure you will too. And that's mostly how we can study bacteria in quote unquote extreme environments, and especially environments that are inaccessible to our current techniques and tools. So space is obviously a great example of that, because we have on the one hand this atypical set of conditions, but we also have to find very clever ways to send them there and also to study their behavior remotely. So Professor A.C. Matin, you've led one such efforts. This was the EcAMSAT mission in 2017, a collaboration with NASA that sent bacteria to space. A.C. Matin is otherwise a professor of microbiology and immunology at Stanford University School of Medicine. And working on bacteria is just one aspect of your work, but he's also have extensive publications on developing exosomes as delivery vectors for cancer therapeutics, and also bioremediation projects via protein and genetic engineering. So it is a great honor to have you speak to us today. I'll give the floor off to you, and I'll remind guests that if they have any questions, they can just type them in the Zoom chat function, and we can do them at the end.
Well, as Ursel just said, I'm not sure I pronounced your name correctly. As she said, please interrupt me anytime you want. I mean, it's not such a formal presentation. And we have an hour about, and I think I can cover much of this stuff. It's 5 after 10, and we will definitely finish by 11. I myself have a few other things to do then, at that time. I'm really delighted that you educated me on so many of my accomplishments. I've forgotten that. How remarkable those achievements are, I don't know. Anyway, we begin with this topic of today, and the title is Earth and Space Research Interface, Bacterial Antibiotic Resistance, and the NASA name for this project is EcAMSAT. They give such names, to microgravity research. And the reason it's an interface is that, the measures that we have discovered are applicable both here on Earth as well as in space, and they're needed in both places. But as I will show you, particularly in space. It's not changing, I don't know why.
At the bottom left of your screen, there are also some arrows potentially, so you could try that.
Oh, yes.
Perfect.
Computer measures. Okay, so you see, this is a work that has been in progress for some time, that took quite a while, and a lot of people have contributed to it. I can't name all of them, but they are listed here. And since you're recording it, you can certainly, investigate who they are and that sort of thing. But if I were to name them, that would be the end of my time. So I'm sure you're aware that antibiotic resistance is a big issue here on Earth as well. And this gives you what you're looking at is the chronology of discovery of different antibiotics. And you can see that, between 1970 and 2005, not that many were discovered. And then you can also see how the resistance are developed. And in fact, by 2005, nearly every antibiotic was subject to being inoperative in bacteria. And at the same time, there has been a dearth of new drugs. And this chart here shows you the number of drugs that had been approved by CDC and other institutions. And you can see only one in 2008 and 2009. And since then, I think the figures are, I don't know the figures, but very, very few. One reason is that, the antibiotic effectiveness lasts only for a short time. Therefore, if you spend a lot of money, pharmaceutical companies, it's only a very short horizon in which they can make a profit, because resistance develops and the antibiotic becomes unprofitable. And there are 2 million people who are infected with antibiotic-resistant bacteria each year in the United States, and several people die as a result of that. So I suspect you're familiar with these things, and stop me if I mention things that you already know, quite well. So here I'm giving you examples of what are called bactericidal antibiotics, beta-lactams that interfere with cell wall synthesis. Penicillin is an example of that. Quinolones interfere with DNA synthesis. Aminoglycoside, gentamicin is an example, interfere with protein synthesis. Resistance to these mechanisms by bacteria has developed, and many of these antibiotics now are largely ineffective. With large doses and all that, they may still work, but it has become a major problem. So these mechanistic aspects of antibiotic effectiveness obviously still exist, but resistance has developed to many of them, and so finding new ways in which you could hurt bacteria by these antibiotics obviously would be very useful. And we have found an additional mechanism, and which is oxidative stress caused by reactive oxygen species. Are you familiar with reactive oxygen species?
Yes.
And the examples are right here: superoxide, hydrogen peroxide. There are many others, And so this offers a new way of trying to overcome the resistance of bacteria towards these antibiotics. And it applies to all of them, as I will show you in a minute. Now, this is another introductory slide, and I suspect, again, you're familiar with it. You know that RNA polymerase transcribes genes. This is the conversion of the DNA blueprint into mRNA. And, there are lots of genes that we all have, bacteria have, certainly we have even more so. And let's stick to bacteria. They don't want to transcribe all their genes all the time. So they want to impose certain specificity on the polymerase to concentrate on the genes that are needed at a particular time. Now, there is the vegetative sigma factor called sigma 70. You're familiar with this, are you?
I think some of the audience would be, yes.
Okay. All right. But the thing is that there are many other sigma factors. And under certain conditions, and I'll just concentrate on one, sigma factor, sigma S, which is called. Yes. So what happens is that under stressful conditions, and I'll give you some examples, its concentration goes up. And how that goes up, we have worked on that, and it's a very elaborate process, and I have no intention of going into it, but its concentration goes up. And as a result, some of the sigma 70 gets replaced by sigma S in the RNA polymerase. And that RNA polymerase then transcribes genes that offer protection against these stresses or disinfectant agents that you subject or one subjects bacteria to. So I give you some examples of the stresses or the deleterious conditions that result in increase in the concentration of sigma S and formation of sigma S RNA polymerase, sigma S species of RNA polymerase that can then transcribe the genes that I will give you some examples of. And here is the example of the deleterious agents that can lead to the increase in the concentration of sigma S and synthesis, and transcription of the genes that the bacterium needs under these conditions. Ethanol, as you know, commonly used antibacterial agent. Starvation, often experienced in bacteria, both in nature and in human body. Low pH in phagocytes, hyperosmosis in so many other places, and many other disinfectants. Any questions so far? Just stop me anytime you want. So some examples of how, of the proteins that are synthesized as a result of the action of sigma S RNA polymerase, which then transcribes the genes, converts the DNA sequence of the required genes into mRNA, and then they are, of course, translated into protein. So molecular chaperones is one example. DNA-K protein family is a very well-studied group. It protects proteins. This UVRABC endonuclease protects DNA. And then trehalose synthesis protects, these enzymes protect membranes. This one, carboxylase, protects cell wall. DSP proteins protect periplasmic proteins, and also several antioxidant proteins that protect against oxidative stress. And I suspect you're very well aware that oxidative stress is a highly undesirable thing. Our mitochondria leak reactive oxygen species. And one of the reasons we grow old is this reactive oxygen species. And again, one of the reasons we develop wrinkles, more so in my case than yours, again, because of oxidative stress. All right, so then you see, what's happening is that these proteins protect biomolecules. And as we have seen, or we will see, antibiotics also act by damaging the biomolecules. So we reason that sigma S should protect also against antibiotics. So this was kind of insight we had. Oh. And this turned out to be the case. Now let's take a look at this ampicillin. You see this, the black squares is the wild type. And you can see that it is not affected by ampicillin. Okay, so this is the top one is the wild type, and this is uropathogenic E. coli that causes cystitis. And the top line here is the bacterium not treated, is the control, not treated with ampicillin. And the lower one with triangles is the culture that has been treated with ampicillin. And you can see that the effect is very, very minimal. On the other hand, if you look at this red curve, this is the mutant missing the gene that encodes sigma S. It's called RPOS. Without the antibiotic, it's perfectly fine, almost as good as the wild type. But if you add treated with ampicillin, very marked death. This is the loss of viability, and it's true of ciprofloxacin, true of gentamicin, true of norfloxacin, and many others. Now, one little note: we have used the stationary phase bacteria for this study for the simple reason that it is in the stationary phase that bacteria live most of their lives. Out there in the environment, there's all kinds of stresses that keep them from multiplying at a exponential rate. They do have exponential rate, but it calms down. It sort of escapes into lack of growth. And inside human body, all kinds of stresses keep them in this stationary phase. And it is in this stationary phase that bacteria elaborate most of their virulent traits. Okay, so the question is, why is the mutant more sensitive to this whole array of antibiotics? And so to determine that, or to have some notion of that, we did what is called two-dimensional gel electroanalysis or electrophoresis of the organelle, the cell extract of the organisms. Are you familiar with 2D gel electrophoresis? Why don't you describe then? You don't want to? Okay, I don't want to put anyone on the spot. Okay, what it does is to separate proteins in two dimensions. One by their electric charge, the other by their molecular weight. And so you can really resolve a whole slew of proteins. Almost all the proteins can be resolved. So now I have to move this a bit, I think. Yes, I can do that. Good. So, what we found that, so you see, we treated the wild type with, I'll concentrate only on gentamicin. It applies also to other antibiotics, but I'll concentrate on gentamicin. We treated the wild type with gentamicin, and we treated also the mutant missing sigma S to see what proteins were not, what proteins were affected in the mutant compared to the wild type. And what we found was that primarily it was antioxidant proteins that the mutant failed to synthesize compared to the wild type. And some examples are given in here, once again, superoxide dismutase and this transaldolase. And these numbers give you the fold difference that was found between the two organisms. So now in the next several slides, I will deal with quite a bit of technical data to show you data which convinced us that GM treatment caused greater oxidative damage in the mutant missing sigma S compared to the wild type. And just to give you a summary, it will be by showing you that genes that are induced by oxidative stress, by reactive oxygen species, they are induced to a greater degree in the mutant compared to the wild type. We use dyes that measure reactive oxygen species, for example, Mplex Red, and it showed greater ROS generation when treated with gentamicin in the mutant compared to wild type treated with the same antibiotic. Damage to proteins was greater in the mutant. It's called carbonylation. And also that protection against GM by antioxidants was very marked in the mutant compared to the wild type. And then also, as I indicated, sigma S controls also proteins of antioxidant defense. So what we did was to also eliminate the sigma S regulated antioxidant proteins one by one, or a couple of them at a time, to see if the mutants missing those antioxidant proteins also became more sensitive to gentamicin. So that's what this is about. Okay. Now, antioxidant proteins like superoxide dismutase or anything else, they require a reducing agent. I mean, after all, you need electrons to be able to quench reactive oxygen species. So you need a source of NADPH or NADH to enable the quencher proteins, the ones that can decompose reactive oxygen species. So a source of NADPH is also required. And if you eliminate that, then again, the mutant becomes more sensitive compared to the wild type. And it is also true in vivo. We use a mouse model with bladder UPEG infection. And now prepare yourself with all of these data. Here is the gene, and you can see wild type. This is okay. There is a way in which you can measure the concentration of the gene by beta-galactosidase synthesis. I suppose you know this, and I won't belabor it. So in the wild type, when treated with gentamicin, the SFIA gene, which indicates exposure of the bacterium to oxidative stress, it is induced, but not that much. In the wild type, it is in the mutant, it is induced to this is the wild type alone. This is wild type without gentamicin, and this is wild type with gentamicin. So there is some increase, correct? But compare it with the mutant. On its own, there is a greater amount of the oxidative stress as indicated by this gene. But when treated with gentamicin, it goes way up. With the dye HPF, you can once again see this is the wild type. It's a very small generation of reactive oxygen species. When treated with gentamicin, it is much greater in the mutant. And this is true of Mplex Red, which detects hydrogen peroxide, and also true of protein damage that is experienced as a result of gentamicin treatment. This is the mutant, it's called carbonylation, and this is in wild type. Any questions so far? And so also, N-acetylcysteine quenches reactive oxygen species. And you can see that, in the wild type plus gentamicin, you have, a certain amount of killing, a very small amount of killing. But when you add this N-acetylcysteine, there is somewhat less killing. But the difference between in the mutant, when NAC is present or not present, is really much greater, as you can see. Both of them are treated with gentamicin. And under anoxic, where oxygen is species, the is just as hardy, as resistant to gentamicin as is the wild type. Well, let's see now. This is wild type plus, yeah, this is the wild type with under anoxic conditions, and this is the mutant under anoxic conditions, and they're nearly equally resistant. And now these are the downstream proteins, downstream of sigma S, the antioxidant proteins that sigma S protects. And you can take my word, in each case, the individual mutant missing any one of these antioxidant proteins was much more sensitive to gentamicin than was the wild type. So here it is, SOD-A, SOD-B. These are again antioxidant proteins, the quenchers. Cat-E, SOD-A again is a quencher. But here, in this situation, these two graphs here, can you see my arrow? Can you see the arrow? Yes?
Yes.
Okay. This ZWF, or you call it ZWF, I'm sure, and GNP. These are enzymes of pentose phosphate pathway, whose main function, one of its main functions, is to generate the reducing power that I was talking about, NADPH. And once again, the mutants missing, lacking the capability of making effective amount of NADPH become much more sensitive to gentamicin. And TEL is another enzyme of pentose phosphate pathway. It's true of other enzymes as well, but I don't have them in here. Okay, and then this SOS response is activated when cells are subjected to oxidative stress. And once again, if you remove sigma S or remove any of the its downstream proteins concerned with antioxidant defense, you find that the mutant is more sensitive. Okay, so this is now in vivo. So what we did here was to inject, we have to practice cruelty in order to be safe ourselves. So we injected female mice transurethraly into the bladder. And we found that GM prevented colonization by the mutant, but not by the wild type. So clearly the mutant had lost its virulence for a mice. But then if you injected the mutant, if you administered the mutant into the mouse with this NAC, the antioxidant, then the mutant became as effective as the wild type in colonizing. So once again, really confirming that antioxidant defense plays a very important role, not only in vitro, but also in vivo. So this is the conclusion. Impairing antioxidant defense will make resistant UPEC sensitive to GM. And this is a little note that we also, in collaboration with many other people, we showed that this is true, even at a single cell level. Okay, so what do you do about that? And this is where the bioinformatics, the little bioinformatics that we have practiced, So we said, if we can, you can't, make sure that people are infected, out there in the wild. When people get cystitis or urinary tract infection, that they be infected with a mutant that is missing sigma S. You've got to find a compound that would silence sigma S or silence, any of the or more than one of its client proteins that act in antioxidant defense. So for that, one of my co-workers, did the virtual screening of a Zinc database of a small molecule, this is the criterion. And so the Uniport database was utilized. This was the software that enabled virtual screening, and then autogrid module and all that. Anyway, so as a result of virtual screening of over 1 million compounds, we found something like 5 that that were quite effective in silencing sigma S and another antioxidant protein salt A. And you can see here this is the structure of sigma S, and this red molecule on it tells you that it is combined to it, and we experiments discovered that this combination impairs the effectiveness of sigma S, and this is another diagram of that. So, especially one compound, zinc was, all these numbers you can see, was especially promising, and its co-administrator effectiveness. Nobody has stopped me. You wanted it to be very informal, but it's sounding more and more formal to me. Anyway, so we go to the next slide. Oh, it worked this time. Amazing. Yes. Okay. Okay, so now a lot of this work was done by, funded by agencies other than NASA, but NASA now and then became quite interested. NASA is just next door to Stanford. The Moffett Field outfit of it. And then they approached us and they said, well, you have shown that this is good on Earth, but what about in microgravity, in space? And this is really an important question because space flight weakens human immune response. This is really quite well established by now. And here is in this arrow, you can see the reasons for it. I mean, not detailed, but some outline of it. Altered immunocyte distribution and function, altered cytokine redox inflammatory balance, and as a result, latent viruses get inactivated. Shingles are often seen, and then clinical incidents. Cystitis is often reported in astronauts. If you go to the NASA website, there is an article in that how much easier it has become now to pee on the International Space Station. I'm not making it up. You can go to NASA website and see. Well, I mean, obviously, urinating on the International Space Station is quite a task. So that's another thing. Okay, so microgravity is an important factor, but there are also others, stresses and this and that, and to tease out as to which one is more important and less important, is a matter that still remains to be determined. So as I mentioned, among the many health issues experienced by astronauts is cystitis. Gentamicin is a standard treatment for this, and that was one reason NASA approached us. So we, along with NASA, asked, would sigma S inhibitors that I just showed you be effective in gentamicin-mediated treatments in space flights? So this was the question that NASA was very much interested in. So, conducting research in space is no joke. I mean, you could do it at ISS, International Space Station, but it's cramped space. People have all kinds of other things to do. So it is important to have an Earth system that can simulate microgravity. And one of the very effective and often used system is this high aspect ratio vessel, HARV, and it generates simulated microgravity on Earth. And so it consists of two kind of vessel or flask or vessels or whatever you want to call them. So when you rotate them perpendicular to gravitational vector, you get simulated microgravity. When you rotate it parallel, it generates Earth gravity. I'm sorry, no. The top one is 1G control. When you rotate it perpendicular to the gravitational vector, you get the control, and the other way you get simulated microgravity. And one reason is that in this situation, the particles, it's a solid fluid phase, and the particles are always falling. That neutralizes the effect. So we examined the effect of sigma S mutation in this simulated microgravity condition, and it showed that indeed sigma S, even under simulated microgravity, makes the organism UPEG, urinary tract, uropathogenic Escherichia coli, more sensitive to gentamicin. Okay, so far so good, but SMG, as you can expect, is only an approximation of real microgravity, and not all SMG findings, simulated microgravity findings, have applied to real microgravity, and I've given some examples in here. Virulence of certain bacteria is affected differently. And in this bacterium, S. typhimurium, whose current name is a bit different, but it is too long, so I left it out. These virulence genes, CYP-T and CYP-C, are affected differently. So the acid test, therefore, is determination in spaceflight experiments themselves to see if indeed silencing sigma S would make gentamicin more effective also under space conditions. Now, for that, yes. For that, so now we are talking about determining GM resistance in the space, and how to transmit the bacterial activity in a space via telemetry to Earth. For you need some kind of a color change reagent. And so as I said, color-based reporter needed to assess UPEG activity for telemetric relay from space. And we chose Alamar blue, which changes color from blue to magenta due to bacterial metabolism. And then the changes, this change in color can be converted into proportional frequency that can be transmitted to Earth via telemetry and converted back to absorbance change on Earth. And this proves to you that indeed the change in Alamar blue from blue to magenta is proportional to the activity of UPEC, the organism we have been interested in. So you can see the wild type is quite active, and the wild type changes the blue into bright magenta. And here it is wild type with gentamicin. It is slightly inhibited, and you can see that the color change is not that intense. This is the mutant without gentamicin. It changes the Alamar blue quite considerably. But when GM is present, gentamicin is present, it is clearly much more blue. Clear? Okay. Oh. So now comes this autonomous microfluidic hardware to test GM sensitivity in the space flight. This really is a very sophisticated piece of equipment, all capable of working autonomously and regulated by radio signals from Earth. And I'm not going to go into all the technicalities, just a few points about it. To determine cell activity, there are these cards used with 48 wells. They're fitted with filters to prevent cell leakage. Air permeable optical quality membranes seal the wells. The card is attached to thermal spreaders, sensors for temperature control. Each well is equipped with 3 colored LEDs to monitor the change in the color of Alamar blue, and then photodetectors for conversion of the transmitted light to absorbance. A bracket is missing here. And then there is the fluid delivery electrically controlled valves. So you see, this is the cell activity part, and this is the fluid delivery part. So electrically controlled valves, diaphragm pumps for high flow rate fluid mixing, precision metering pumps to deliver reagents, for example, gentamicin, the medium and buffers and such. And this is you have a question? I thought somebody raised a hand. Okay. So this is the fully assembled EcAMSAT biological, fluidic, optical, thermal payload system right up here. And B is its hermetic payload containment vessel with electrical interface board. And all of these are really highly sophisticated bits of engineering products. And this is a little bit more detailed of it. Passive magnetic orientation. Once it is in the space, it orients itself to the Earth's magnetic field. Ultra-high frequency beacon and S-band radio, extensive thermal power management, testing and modeling. And then you see the signals that the EcAMSAT sends from the space were monitored at Santa Clara University, which is also in the neighborhood, who have these big what are they called? The the name is escaping me. These round thing that capture signals. The name will come back to me. Anyway, so the flight experiments were programmed to start autonomously. Daily radio communication with this spacecraft were conducted, and Alamar blue absorption was transmitted via telemetry, as I've already mentioned. Okay, so you see, this EcAMSAT is part of NASA's, development for carrying out space experiments without having to, to impart on them self-flying and self launch capability, and therefore it makes it much cheaper and much more effective for carrying out space experiments. And so you can see, oh, I'm sorry, I'm going the wrong way. Okay, so it was launched on an OUBT mission on November 12, 2017. So a rocket was going to go to International Space Station, carrying many other things, but this thing was piggybacked, as they say, on it. And it reached the International Space Station and was deployed from there on November 2017. And this is it. You can see in front of the arrow coming out of the space station into the microgravity atmosphere. Okay. And these are the results. This is the telemetric results transmitted by that from Santa Clara University by the name is still not coming. Anyway, so these are the details, but these are the results, and you can see that wild type. This is the survival in flight. The wild type survives long. These are both treated with gentamicin. This is the wild type, and this is the mutant. So again, in space flight, the mutant is more sensitive, and therefore the small molecules that we have discovered and that have to be tested further, offer the promise of enhancing the effectiveness of antibiotics like gentamicin, and as I mentioned, is true of many other antibiotics as well. Now, the last question that was raised as to how you enhance the survival of beneficial bacteria. But this is a stuff that is not my area of expertise at all, but quite a lot of work is being done for this, or in this area. And, but it is, way in the future. As far as the space station is concerned, this is the replica of they have there now. And most of it is mechanical, CO2 removal by scrubbing, oxygen by electrophoresis, and by cylinders from Earth. But these closed systems that people are trying to establish would have the capability of waste recycling and purification, providing food and oxygen, microbes to break down wastes, technology to remove impurities from air and water. That's it.
Great, thank you so much. You gave us a lot of background which was helpful at the start. So I'll just give some time for people to type questions in the group chat, and then while I do that, I can maybe start with some of my own. So, I mean, definitely when you're studying bacteria, a big question is always individual bacteria versus group behavior. So when bacteria assemble in biofilms, do you expect that would influence your results?
But you raise a very good question. And in fact, we have studied biofilms as well, but entirely in the simulated system using those halves. And, once again, as you know, biofilms are more resistant than individual bacteria, orders of magnitude at times. And by and large, sigma S is useful there as well.
You have a question that says, what experiments are going to be conducted with microorganisms and bacteria in space, and what's their behavior in space? So maybe the question is regarding future experiments.
Future experiments is a huge, okay. Look, this entire study is in a state of infancy. It's not easy to conduct experiments in this space. And for instance, you see this EcAMSAT system that I just showed you. It has its own problems because you see, it subjects bacteria to many different kinds of stresses. And remember, I mentioned sigma S is the key to respond to stresses. So you have already stressed the bacterium, in a system like the EcAMSAT, and then you are exposing it to a microgravity, which is another form of stress, and then to antibiotics, additional stress. So how you tease these things out is not that easy, right? So, and in fact, if you look at the literature concerning bacterial behavior in space, you see that there are quite a few contradictory findings. Some people have found that bacteria become much more resistant to antibiotics. Others have found that they don't, that the lag phase changes or the exponential phase changes or the amount of biomass that is generated per amount of food consumed increases, and others have found that that is not the case. And in fact, those conclusions may be based on certain artifacts. So the short answer is that this is just the beginning of these studies. And those of you who are interested in microgravity, you have a fertile field ahead of you. And as I told you, I was at Oxford 2 years ago, and there was a complete audience, speakers as well, of course, very much interested in microgravity and the commercial aspects of it. So it's another way of getting rich.
Yeah, I mean, you mentioned there being a lot of contradictory findings in the fields. So was your paper well received, or and have people tried to reproduce it since and use the same apparatus for their own experiments?
You're asking me whether our paper was well received? In
part, yes.
Yes. I just wanted to be sure it was extremely well received. if it was the selected article, both of them, the one that preceded it, which described the EcAMSAT on Earth itself, just to show that it had what capabilities it had. That, as well as the flight experiment, were very well received. And the reason is, you've got to judge things in relation to what is possible. I mean, you can't achieve perfection right from the very beginning. So we acknowledge that, in fact, in the paper, if you take a look at it, we do say that, it is difficult to exactly tease out. Nevertheless, the bulk of the evidence strongly suggests that this sigma S silencing would be helpful.
Great. Maybe Rocky, you have a question. You can either unmute yourself or post it in the group chat, whichever you prefer.
You're asking me or someone else?
Someone has raised their hand. Please feel free to unmute and ask the question. Rocky.
Please speak slowly. I think your voice is not coming. I hope mine is coming clearly to you. Is it?
It is. It might be the Wi-Fi. So someone had a question, but I don't think they've that it's going through. So maybe I'll ask one last one before, because we are reaching the hour.
Yes.
Your experiment was it was weeks, so it was a relatively short time scale. But in very long spaceflight conditions, I suppose E. coli would have thousands and thousands of generations to adapt. So do you plan to replicate these experiments, but for example, on a much longer time scale to see if some different behaviors would emerge?
But, I don't intend to myself because, I've got other projects as well. My main interest these days is cancer therapy, using prodrugs and things like that. But I think that in the future, definitely, NASA will continue with this. I mean, remember the sorts of things that have to be investigated are huge. I mean, the effect of microgravity on human bones, the possibility of growing plants, in a good way, in a sufficient way, ensuring survival and proper activity of bacteria that you need for recycling of waste. These are all complicated questions. And NASA has got to, deal with all of them. And before you can ever hope to be able to colonize anything in space, any planet in space, a very hostile environment, as I'm sure you know. So yes, the short answer is that yes, this work will continue, but whether I'm going to be doing it, the answer is no.
Okay, and I have a very wild question in the group chat to finish it off. Is there a possibility that our genes can be stored, and in a future where a mission moves to Mars, then we can just generate the body there? I don't know if you have any thoughts on that.
We are very much into futurism here, aren't we? I mean, I told you that it's really, at the moment, it's a pipe dream to have a colony in space. I mean, but remember, we have had many pipe dreams in the past that have come to fruition. I mean, imagine 200, 300 years ago, the idea that you could fly from one place to another, thousands of miles away, would be possible. So all of these things, I think, are possible. But your question as to whether we can save our genes and, because it takes a long time, right, to fly. I mean, you're talking of light years, certainly not as far as Mars is concerned, but exactly what distance do you have in mind about the gene situation?
I mean, the person who asked the question said Mars. So
Mars.
I suppose that's, in the solar system scale, but it is speculative.
But why would you want to save the genes in order to go to Mars?
Maybe Space Infinity, do you want to expand on your question?
I'm sorry, you have?
No, I think it's fine. We can keep it food for thought because we are approaching the hour. Maybe I can invite the Space Infinity, the guest, to email you.
Please do that. I think what you should do, if you are interested, you can consult your colleagues, other people who may be interested in this, and put your questions in writing. Just email those to me. You have my email, and I'll give you answers as best I can.
Great. Well, on that, I'll thank you again for speaking to us. It was really great talk, and thank you to everyone who attended. And as I said, I think Professor Matin would be very happy to answer any questions. So you can email him with any follow-ups that you have from this talk.